Hello, I am Ricardo and I am a nerd scientist. I work with other nerdy peoples in a pretty nerdy place, Karolinska Institute, a research excellence in Sweden. As all the other geeks, I also get pretty excited about my research. I work with bacteria, those tiny creatures you might remember from high school or from some yogurt commercials. You might also know that trillions of these little animals lives inside you. They protect your intestine, digest your food, and produce your vitamin. Wait, there is more. Recent data also show that bacteria can control your cells. They modify your DNA, and they can control your brain and cognitions. That's pretty awesome. Bacteria might also decide whether you're going to develop some chronic disease. More than 1 million of United States citizens suffer from the inflammatory bowel disease. 2 million are in Europe. How do bacteria in your intestine decide if you're going to develop a disease or not? We have an idea. We think that some bacteria that lives inside you carry a weapon, a powerful gene that contains the information to create a toxin that might potentially be used to shoot directly into your gut. We think that this weapon gene might be a tool by which some bacteria decide and control your health. To test this idea, we took bacteria from the gut of healthy people and people who developed some chronic illness in the gut. Right now, we are looking for the weapon gene inside these samples. Now, to look at a gene, my naked eyes should be 1 million times more powerful. 1 million. I can squeeze my eyes a little bit, but that's way too much. How do a scientist look for a gene?
DNA is a long novel. In this novel there are thousands of sentences composed by millions of letters. We could read this novel exactly how we read the book, but these letters are way too small. Genes are the sentence that compose the novel. To be or not to be, and this is the question, might look like DNA gene. Our weapon gene would look like, I'm a bad gene and you will never find me. Well, we will manage to find it if we use the right tool. To catch a specific gene, we need the help of a special Google enzyme, properly named as polymerase, plus a couple of queries. Imagine that you want to find out the lyric of a song online, but you remember only a few words. We put everything in a small tube, we shake, heat up, and this is what happened. The two strings match perfectly the gene, so the polymerase enzymes see them and start to copy the gap between the two. 1, 2, 3 times. Since the polymerase is very stable, this copy machine system can potentially go on forever. Scientists have the control and can decide to stop the reaction when they like, or when the space on the board is over. We have just amplified the original gene and obtained billions of copies. So a scientist might not see one gene, but when they become billions, it's hard not to notice them.
This experiment is at the very base of great human discoveries, but it's very expensive. We are exploring how the smallest living creatures on this planet influence human life with an incredible laboratory technique, and you can be part of this. Become a backer and you will follow the progress of the project, have access to the behind the scene of the lab, and get our exclusive rewards. The answer has been already written in Nature, we just have to read it.